This is for Biology 223, genetics. This is an article, a short research article. It's called a letter, published in Nature. It's published in 2014. And the title is An Evolutionary Arms Race Between KRAB Zinc-finger Gene ZNF91 and 93 and SVA/L1 retrotransposons. So what caught my eye here is the concept of evolutionary arms race. We have the normal meaning of arms race between military powers, where one develops a military technology, the other one responds, they compete until, well, bankruptcy, like the Soviet Union or whatever. And we have an analogy in evolution between host and pathogen, where a pathogen has a mechanism of entering the host or parasitizing the host or causing disease, and the host responds, and then the pathogen responds to that. And so we have examples of pathogens in humans, for example, that have a mechanism, the host responds, let's say, through the immune system, and the pathogen exploits that immune system response into the cell, whether by, for example, infecting a macrophage as a means of entry. So those are quite common and of great interest in understanding how evolution occurs and what sort of genetic mechanisms and how different systems exist because their exploitation reveals them. So throughout evolution, primate genomes, primates include us, primate genomes have been modified by waves of retrotransposon insertions, right? A retrotransposon is integrated in the chromosomal DNA and then is expressed as an RNA and then reverse transcribed into cDNA, and it can be reinserted at a different locus. And so we have these waves of retrotransposon, and so the host develops mechanisms to suppress retrotransposons, and the retrotransposons evolve ways to escape that. Whether a random mutation, they escape, they can leak. So we have been modified, or genomes have been modified by these insertions. For each wave, the host eventually finds a way to repress retrotransposon transcription and present further insertions. And you might ask, well, what happens if it doesn't find a way? It goes extinct, or that germ, that lineage goes extinct. And so for each wave, the point here is the survivors have found a way to repress transcription. In mouse embryonic stem cells, which is a good model for this, transcriptional silencing of retrotransposons requires CAP1, also known as TRIM28, and its repressive complex, which can be recruited. So it requires these silencing and the recruitment of these genes, these factors, are recruited by KRAB zinc-finger proteins, KZNF, such as murine-specific, so ZFP809, which binds the integrated murine leukemia virus DNA elements and recruits CAP1 to repress them. So that's a phenomenon. Still, we have a phenomenon. We have description of what happens, and we haven't come to the question yet. KZNF genes are one of the fastest growing gene families in primates, and this expansion is hypothesized to enable primates to respond to newly emerged retrotransposons, newly emerged over millions of years, hundreds of thousands of years. And we have hypothesized, so we may have the gap in knowledge yet. We may not have the gap in knowledge yet, but we have a hypothesis. However, the identity of KZNF genes battling retrotransposon currently active in the human genome. Current is over a long period. So the gap in knowledge is we don't know which KZNF genes are that are battling the retrotransposons that are active in the human genome currently, such as the SINE VNTR ALU, those are the SVA. This is what's used for VNTR fingerprint analysis of people. There's a lot of variation in the human gene pool of this. These are short interspersed nuclear elements. They're parasitic on the line ones, which are the long ones. So these that are active, most commonly retrotransposing in the human genome, are these SVA, and the long interspersed nuclear elements, line 1 or L1. So we have L1, SVA. That's what's active now, and that's what the question is. Here we show that so the identity of the KZNF genes battling SVA and L1 retrotransposons currently retrotransposing in the human genome is the question. Here, so their results, their findings, we show that 2 primate-specific KZNF genes rapidly evolved to repress these 2 distinct retrotransposon families, right? Those are unrelated or distantly related families, shortly after they began to spread in our ancestral genome. Now remember, we're seeing only what exists and has not become extinct. So remember that bias here. We're finding out, we have access to what worked. So, and we have in our own genomes the history of retrotransposable elements and the genes active against them that will be in the record of our genes and our chromosomal sequences. ZNF91 underwent a series of structural changes 8 to 12 million years ago that enabled it to repress SVA elements. ZNF93 evolved earlier to repress the primate lineage line 1 lineage until 12.5 million years ago, so not current, when the L1PA3 subfamily of retrotransposon escaped ZNF93's restriction through the removal of the ZNF93 binding site. So these ZNF proteins recognize a sequence, and that's how they target this CAP1 to silence them. And so if by mutational pressure the retrotransposon here loses the binding site, it escapes. Our data, so now we're going to have the interpretation, the significance. Our data suggest a model where KZNF gene expression limits the activity of newly emerged retrotransposon classes, and this is followed by mutations in these retrotransposons to evade repression, a cycle of events, recurrent cycle of events that could explain the rapid expansion of lineage-specific KZNF genes. So they have a nice model here, and what struck me about the paper, it's another example of retrotransposons related to chapter 7, of regulation of gene expression in eukaryotes, that's also a text chapter, and also the sort of things that are involved in tumorigenesis, that's also a chapter on genetics of cancer. And the work is particularly elegant in that they have a very nice figure here in which they have parallel the evolution in the ZNF93 throughout our lineage. Humans are closer living relatives, chimpanzees, and then including the gorilla and other great ape, orangutan, and then other ape, gibbons, so and old world monkeys, I believe that is. So throughout this lineage of our ancestry, one can see that there's evolution of the ZNF93, that is the gene involved here in responding to retrotransposons, and that parallels the evolution of the retrotransposons in our lineage. So we can mirror that, right? The evolution of the retrotransposon through time and the evolution of our response. And we don't just have our own genome to understand that. We have the genome of relatives in the old world monkey, apes, great apes, and there's all sorts of specialized name for the subfam tribe and subfamily and family. And that is also seen in the case of the short interspersed nuclear elements here, the SVA. We have a very similar pattern. So it's a really, really nice way to show this, and the work has nice way to correlate the big picture to specific concepts in deep sequencing and ways to express binding sites and their mutation and consensus. So I want to emphasize that this is a very nice example from 2014, though, of the sort of work that's at the forefront in genetics. I will stop here.